Dear students, Module 14. Structural chromosomal aberrations. Under that, deletions with suitable human examples. The main objectives of this episode are to know about the chromosomal abnormalities and their consequences, to explain the structural chromosomal aberration, to understand about the deletion and its classification, to know about the disorders that are caused by the deletion, to explain the causes for the chromosomal abnormalities. Introduction. Chromosomal aberrations are one of the essential biological consequences that arise by human exposure to some ionizing radiations and other genotoxic agents. They also act as the primary cause of many human genetic diseases and other abnormalities. Currently, they are the underlying causes of the high percentage of spontaneous abortion during the early pregnancies. People with high frequency of chromosomal aberrations in their peripheral blood lymphocytes have a high chance of developing cancer. Thus, many types of cancers are associated with specific types of chromosomal aberrations. These chromosomal abnormalities or aberrations can be numerical or structural. They can affect autosomes or sex chromosome. The phenotypic consequences of each chromosomal abnormalities rely on the nature of the abnormalities, specific chromosome involved, genes affected, and finally, the likelihood of its transmission from one generation to another. The structural abnormalities results from the loss or gain of the chromosome structure or segments. This will mainly occur when parts of the chromosome is missing, or part of the chromosome extra, or part of the chromosome is transferred to other places. These aberrations can be balanced or unbalanced. In case of the balanced condition, the rearrangements can happen that does not produce any gain or loss of the chromosomal segments, and hence it is harmless, which do not produce any abnormal phenotype. The unbalanced condition can lead to chromosomal complements that contains incorrect chromosomal material due to the loss or gain of chromosomal segments. These conditions result in clinical effects are usually severe. All the conditions of structural chromosomal aberrations can cause some congenital disabilities. Hence, in this episode, the following aspects are going to be discussed. A. Structural chromosomal aberrations. B. Deletion as the structural chromosomal aberration. C. Examples of deletion. D. Causes and consequences of deletions. A. Structural chromosomal aberrations. The changes that occur mainly in the chromosome structure is structural chromosomal aberrations. These structural changes can occur due to the breakage or joining of other structures that are different from the original chromosome complement. These abnormalities are due to the changes in the number and the order of gene, not because of the mutation. Structural chromosomal aberrations are mainly classified into deletions, duplications, translocations and inversions. Deletion is the loss of chromosome portion due to the breakage that occur one or more regions on the chromosome. Duplication is the process where the chromosome is duplicated, which result in the formation of repeated segment of genetic material. Translocation is where the movement of the genetic material or the segment of the chromosome takes place between the non-homologous chromosomes or within the same chromosome. Inversion is a process when there are two nicks or cut on the segment on the chromosomes, they get inverted and reattached. All these structural abnormalities in the genetic material will disturb the whole genetic composition and can lead to the chromosomal instability syndromes. These are the groups of disorders characterized by the chromosomal instability and breakage. B. Deletion as the structural chromosomal aberration. Loss of chromosomal segments is lead to the loss of genes present in the missing segments or portions on the chromosome. A large number of deletions can be lethal in case of monosomic conditions. Hence, with the missing of a large number of genes, about 2% of the genome leads to the genetic imbalance that cannot sustain life. The spontaneously occurring deletions must include 2 breaks in the intervening segment on the chromosome. If 2 ends after the breakage get joined, then they show a shortened chromosome with centromere. The deleted or lost small segment is said to be acentric without centromere. This resulted in the acentric fragment is immobile and will be lost. Chromosomal anomalies in human embryos that are produced by in vitro fertilization are common, and they can include structural abnormalities along with numerical aberrations. Studies have identified that the deletions are the most common structural anomalies, accounting for approximately 8% of the euploid blastocysts. Further, research on the 15 euploid blastocysts using microarray platform and mapping techniques, it was revealed that the deletion of about 81 to 464 genes, and out of these genes about 34 to 149 genes are related to some known genetic diseases. Hence, the loss of many essential genes can take place that can cause genetic problems. Some other lost genes were related to developmental delay and intellectual disabilities. Thus, it is crucial to consider that the embryos with chromosomal deletions revealed by the microarray should not be transferred to the patient. Further, gene mapping or phenotype seeking is necessary in those cases. Deletions are mainly classified into terminal deletion, interstitial or intercalary deletions, and micro deletions. Terminal deletions is where the chromosome end is get broken, loss of the terminal part of the chromosome takes place. This can occur by one cut towards the end of the chromosome. In case of intercalary or interstitial deletion, two breaks occur to delete the interior part of the chromosome. Hence, pairing of homologous chromosome during meiosis loop can be seen. Microdeletion is the loss of a tiny segment about 5 MB that can include few genes. This type of deletion is usually found in children and some physical abnormalities. Mainly, the effects of the deletion depend on the size the chromosome lost. Depending on the size, deletion can be intragenic and multigenic deletions. A very small deletion within a gene can inactivate the gene and gives the effect the same as other null mutations. These small deletions within the gene are referred to as intragenic deletion. This deletion can be distinguished from the single nucleotide changes because they are non-revertible. The consequence of this intragenic deletion are mild. The homozygous null phenotype in case of human albinism is a viable condition. Hence, this can ensure that the homozygous deletion will also be viable. In case of multigenic deletions, it is involved in the loss of 2 to 1000 genes. Hence, this type of deletion can show severe consequences. By inbreeding, such deletion is made homozygous when both homologs have the same deletion. This combination is always lethal. This will reveal that most regions on the chromosomes are very important for normal development and viability. Formation of deletion loop. Sometimes deletions can be identified by the cytogenetic analysis, but by the formation of deletion loop, they can be easily recognized during the examination of meiotic chromosomes. When an individual carry a heterozygous deletion, a region of the deletion can be determined by the failure of the corresponding segment on the normal homolog to pair. This will result in a deletion loop. Deletion loop can be observed in case of Paulitin chromosome of Drosophila fly, where the homologs are fused. The genes in this deletion loop will have no alleles to synapse during the meiosis. Since Drosophila Paulitin chromosome consists of specific binding pattern, it becomes easy to infer that which bands are missing from the homolog by observing the bands appeared in the loop of the normal homolog. Genetic properties of deletions. The genetic criteria are very useful for some species whose chromosomes are not easily recognized by cytogenetic analysis. When the chromosomes carrying deletion are homozygote, then the cells fail to survive. This situation can also occur if there are any lethal mutations. Reversible conditions of chromosomes is seen when they carry a deletion can never revert to normal condition. This genetic criteria is useful if the organism shows some specific phenotype associated with the deletion. When the chromosome carrying deletion is a heterozygote, the recombinant frequencies between the genes that are flanking the deletion on the chromosome are lower than the control crosses. The unpaired region on one of the homologue cannot participate in crossing over. See examples of deletion. Deletions are regularly finding in human chromosomes, and in most of the cases they are relatively small. But the effect of small deletion shows adverse phenotypic changes even in heterozygous condition. Deletion in specific chromosomes lead to unique syndromes with many phenotypic abnormalities. One of the example is Cray-Duchat syndrome. This is mainly caused by the heterozygous deletions of the tip of the short arm of chromosome 5. Hence, it is also known as 5p minus syndrome. The size of the deletion varies among the people. An individual with small deletion can show mild effects, and larger deletion can show severe intellectual disability along with developmental delays. A child with this syndrome has a unique character of high-pitched crying that sounds like that of a cat. This syndrome can occur in about 1 in 20,000 to 50,000 newborn infants. This condition has been found in people of all ethnic backgrounds. This condition is not heritable. The affected individual will not have any family member with the same condition. The condition can occur randomly during the formation of reproductive cells. Many of the symptoms of this condition are because of loss of multiple genes. Researchers have found one of the genes called CTNND2 5p15.2 deletions showed association with the severe intellectual disability in some individuals. Some of the symptoms of this condition include microcephaly, weak muscle tone, developmental delay, low set ears, small jaw, widely set eyes, problems with language, and express themselves with numbers or signs, hyperactivity along with the heart defects and intellectual disabilities. Wolff-Hirschhorn syndrome is caused by the deletion of short arm end on chromosome 4, 4p minus. In this condition also, the size of the deletion varies from affected person to person. Larger deletion can show severe intellectual disabilities and physical abnormalities. Some of the genes have been identified related to some of the typical symptoms. They include NSD2 4p16.3, LETM1 4p16.3 and MSX1 4p6.2 genes. NSD2 deletion is responsible for many features of this condition, including some facial abnormalities and developmental delay. LETM1 is associated with seizures and abnormal electrical activity inside the brain, and MSX1 is associated with dental abnormalities along with the formation of cleft lip or clipped palate. Wolff-Hirschhorn syndrome occurs in approximately 1 in 50,000 newborns. It is also observed that the condition affects females twice than males. Some of the facial abnormalities include flat and broad nasal bridge, wide forehead, cleft lip or palate, small chin, poorly formed ears with small holes and small head, etc. Jacobson syndrome is a rare condition caused by the deletion of genes on the long arm of chromosome 11. The prevalence of this condition is estimated to be 1 in 1 lakh newborn infants. This condition is also not inherited and mainly is associated with physical defects. Some of the facial symptoms include wide set and droopy eyes, broad nasal bridge, low set ears, small lower jaw, macrocephaly, trigonocephaly, which is the pointed look of the forehead. All these three syndromes can be caused due to random deletion and vary in size. In a few cases, the affected child can be born from an unaffected parent with some rearrangements in the chromosome that is not harmful and will not show any phenotype in the parent. This is called a balanced translocation, but it can also arise spontaneously in the germline of the parent. When it inherits to a child, it can get unbalanced and cause harmful effects. This is called as unbalanced translocations. In those cases, parents should be cautious to have the extra baby because parents will have a high risk of having another child with the same condition. One more example of the deletion can be observed in Drosophila, where it shows that the deletion can produce some unique phenotype. The fly shows unique notch wing phenotype when one of the homologs carry the deletion. Hence, this deletion show a dominant mutation, but the deletion is lethal in homozygous and therefore act as a recessive in regards to its lethal effects. The specific dominant phenotypic effects of some deletions can be caused by the chromosome break inside a gene which, when disrupted, will act as a dominant mutation. In the case of Notch locus of Drosophila, it contains both sex-linked dominant and recessive visible mutants. Hence, they can show pseudo-allelism. D. Causes and consequences of deletion. Some of the mutagens, including ionizing radiation, X-rays, gamma rays, etc., can induce the chromosomal rearrangements. These ionizing radiations are highly energetic and are enough to cause the chromosomal breaks. Depending on the breaks produced on the chromosome, different possible deletions can occur on the chromosome. There are also some external factors including heat, viruses, chemicals, and some errors in the recombinations can cause deletions on the genetic material. The complete elimination of the genome fragments is deleterious. Even in the case of heterozygous condition consisting of one homologue which is normal, another with deletion may not survive. First, this can be by the imbalance of genome by the large deletions of genes. Second, in many organisms there are recessive lethal and other deleterious mutations throughout the genome. The wild type alleles can cover these on the other homologue and this may be expressed. But in case of deleterious conditions, there can be uncovered or unmasked recessive lethal alleles and they can express to give phenotype. The recessive allele showing the dominant effect is termed as pseudodominance. Haploinsufficiency condition can be seen where the product of one gene is not enough to produce the normal phenotype. To conclude, deletion is the common structural chromosomal aberration seen in many organisms. Apart from its effects, these can be used currently in much genetic research to understand the functionality of the deleted gene or genome segment. Thank you.